to the hot springs, as the name suggests, the temperature in the hot spring is going to be a lot warmer than any lake or ocean or sea. And so these are the native habitats of these fish, and it gives you the range of temperatures in which they're living from -2 Celsius for the Antarctic, as opposed to 32 to 42 degrees Celsius for the hot springs, which is much warmer in Celsius. So that's a lot warmer than what you get in the Antarctic. So the range of the temperatures is given on the x-axis. And on the y-axis, you have what we call the half time for the enzyme to denature. So when you take the enzyme from the animal and put it at 37 degrees, how long does it take for the animal? And so the numbers you see above each of these columns is the number of species they studied. So they only studied one species from the hot springs, only one species from East African lakes. They got 8 species from the Indian Ocean. They got 8 species from the Mediterranean. They got 6 from the North Sea and 3 from the Antarctic. So the number of species is not that great, but it is showing you some interesting information. So when the fish is normally living in the Antarctic, where the temperatures are -2 to 2 degrees Celsius, and you extract the same enzyme, which is a muscle enzyme, myofibrillar ATPase from fish, and you take that particular enzyme, and then you look at the activity of that enzyme at 37 degrees Celsius. So that is standardized. So all of the enzymes are studied at 37 degrees. How long does it take for that enzyme to denature, for half of the enzymes to denature? And you can see that it doesn't take much time at all. So in just a few minutes, at least half of the enzymes have denatured that were extracted from these fish. When you look at the enzymes from the fish from the North Sea, it takes a little longer for them to denature. If you look at the ones from the Mediterranean, there's a much larger range, and on average, it takes a lot longer to denature. And as you go along to the right, you can see the hot spring enzymes. You're looking at 600 minutes here, and they're just beginning to denature. So they last a lot longer at 37 degrees, which should make sense because the temperature at which the fish normally lives is 32 to 42. So it's smack in the middle of their normal temperature range. And so their enzymes are very, very stable at 37 Celsius. Whereas the enzymes from the fish from the Antarctic are not stable at 37 degrees because they're not used to living in those temperatures, and their proteins denature very quickly at high temperatures. The interesting thing is that it's the same enzyme. So what is different among all of these fish that the enzymes from some fish denature so quickly at 37 degrees, and the same enzyme from other fish last a long time at 37 degrees? Yes, sir.
The temperature at which they're not the temperature at which their natural habitats.
So something about the temperature at which they normally live has selected for these different enzymes, but they're the same enzymes. So what is it about the enzyme that has been selected for that makes them stable or unstable? Yes, there was selection due to the natural habitat. If you happen to have this enzyme, if you vegetate this fish and throw it in the hot springs, they'll die. Because that enzyme will denature, most of its other enzymes will denature, it will die. But it's the same enzyme. So how come the enzyme in this fish doesn't last at 37 degrees and the same enzyme from those fish lasts a long time? What is different? Yes, sir. Something about the shape that is remains stable. So what is it that makes this enzyme stable and that one not stable at 37 degrees? Yes, ma'am.
The hydrogen bonds are stronger.
The hydrogen bonding is different. So what allows for differences in hydrogen bonding? I mean, hydrogen bonds are going to break as the temperature rises, but what is going to change the folding pattern of the hydrogen bonds of the enzyme? If they're the same enzyme. Go back to the protein structure. What are the different levels of protein structure? Primary, secondary, tertiary, quaternary. So what has to be different?
The primary?
Primary structure has to be different. And what makes the primary structure?
Amino acids.
The amino acids. So yes, they're the same enzymes, but they must have differences in which amino acid is where. Which means there is variation in the enzyme in the different fish. So the primary structure can't be the same, which causes differences in secondary and tertiary structure, which means that some of the enzymes are stable at warmer temperatures and some of the enzymes are not. So this is at the molecular level what you're looking for when you say there is variation in the population. So in the fish populations around the world, same enzyme has variation in it. So there are differences in the amino acids, in the sequence of the amino acids. And that means that the shape and the structure of the enzyme will be slightly different, even though they're the same enzyme and they do the same thing. And so natural selection, through the temperature changes in these environments, could eliminate some and favor others. So if you were to have the wrong sequence for the enzyme and you happen to be born in the hot springs, you might die because you just have the wrong sequence, your enzymes won't function properly. And then you will be eliminated from the population. So far, so good? Now, will this variation be heritable? Yes, why?
Because if your enzymes are denaturing, I guess at a different and quicker rate than the fish that will be able to live long enough to.
You're talking about differential reproduction and survival, so we'll get to that. I'm asking you whether this variation among the enzymes, having different protein sequences, would that be heritable? Yes, ma'am. I can't hear you at all. So the point that she's making is that any amino acid that's in the protein was coded for by the gene sequence in the DNA. Therefore, it's heritable because proteins are coded for by DNA. So if you have a difference in the protein, that means there must have been a mutation in the DNA that caused that change in the protein. So all the variability that we see in the same enzyme in all of these fish was caused by mutations in the DNA, changed the sequence of the amino acids, therefore you got differences in the protein. Sir, you can't sit in the staircase area, you're blocking the exit. So it is heritable. So there is variation and it is heritable, and then you can talk about differential survival and reproduction, that if you had all of this variation within the same environmental area, if all of these various fish were present, let's say in East African lakes, where the temperature range is 20 to 30 degrees Celsius, then some of those fish would die because they would have the wrong shape, wrong type of protein with wrong type of amino acid sequence. They would not be able to function properly, they would die. The ones that would survive would then reproduce, some of them better than others, and then those inheritable, the heritable variation would then be passed on to the next generation. So you should be thinking of those lines of logic when you take a look at examples like this one. So here we're looking at a different enzyme, again from the muscles of fish. And this is a North American fish genus that is studied in lakes going from southern North America to northern North America, and it's present in all over the place. And so they looked at different versions of this particular enzyme, and they found two different types. So there's the A type and the D type. And the A type produces the enzyme that is, has an optimal temperature at 30 degrees Celsius, and the D type produces an enzyme that has an optimal temperature at 20 degrees Celsius. And when you look at populations in the south, they tend to have the A type, and as you move further and further to the north, you progressively get more of the D type in the population.